The topic I'm going to address relates to a fundamental sea change that is happening in the field of age-related neurodegenerative disorders, and it comes under the catch cry or phrase prion-like processing of abnormal proteins. And basically I want to show you that that is the case, or very likely to be the case, and secondly, that it provides a basis for a unified pharmacological approach as well. So the basic concept is very simple, that proteins are made on ribosomes from RNA, and that they fold in some complex primary structure and then form complexes. What happens in the disorders of protein aggregation is that for reasons that are unclear, although are becoming increasingly clear, the unfolded protein either begins to form an aggregate, often with a beta sheet core, or a disordered aggregate. And this is really the danger. Once it forms these aggregates, a clearance pathway is no longer available to it. Now, the surprising thing that's emerged in the last few years is that in all of these cases, the pathology spreads through the brain, most likely via an infectious process. So in Alzheimer's disease, which I'll talk more about, we have the beginnings of pathology in the medial temporal lobe structures, and actually earlier than that in the basal midbrain. And then it spreads into isocortex. But it turns out that exactly the same thing is going on in Parkinson's disease and in Huntington's disease. The mechanisms of spread are not known as yet, but various theories are developing. So I'm going to go through tau protein, which is where my main expertise lies, but I'm also going to then talk about the other proteins as well. So what is tau protein? Tau protein is a microtubule stabilizing protein. So it binds via repeat domain to microtubules, and it's the axon-specific microtubule binding protein. These projection arms space out the microtubules, and if you have a tau knockout mouse, then the microtubules bundle. But it's more important, the function is more important than that. The microtubules, and therefore tau protein, assist in the transport of synaptic vesicles from the cell body to the synaptic terminal. There are different isoforms of tau that depend on alternate splicing at two sites in the N-terminus, and a very important site in the repeat domain that results in the formation of either a 3-repeat or a 4-repeat isoform. There's a growing body of evidence that mutations, particularly around the repeat domain of tau, are associated with primary neurodegenerative disorders, particularly the so-called FTD-P17 class of frontotemporal dementia with Parkinson's disease. So tau protein, as I say, normally stabilizes microtubules. Its role in the axonal communication pathways is critical in the pyramidal cell system, which communicates different regions, different cortical columns in the same hemisphere or in the contralateral hemisphere. So ultimately, this aggregation of tau protein is a disorder of the pyramidal cell system of the brain. So the aggregates begin as oligomers and progress to protofibrils and form fibrous aggregates, and ultimately are left behind. So these aggregates are what Alzheimer in fact discovered and called tangles or neurofibrillary tangles, and that's why the disease is called Alzheimer's disease, basically because of Alzheimer's discovery of the tangles. If we look into these tangles in greater detail, so here they are in the histological section. If you pull out one single tangle, this is where the nucleus used to be, you can see it is a dense array of these characteristic fibers called paired helical filaments, or PHFs. We initially solved the structure of these filaments and showed that at a low resolution it's two C-shaped subunits and it forms a polymer. and it's coated on the outside with phosphorylated tau. When you do a study of the mass and the biochemistry, people often say in literature that paired helical filaments are composed almost entirely of phosphorylated tau. That's not the case. Less than 5% of PHF tau is even full length, let alone phosphorylated. So the phosphorylation bit is added almost as an afterthought or as a late stage in the assembly process. If you look deeper inside at the core of the paired helical filament, and we did this when we first isolated tau from tangles and showed that it's the critical constituent of the paired helical filament, we found actually 3 fragments. So here's the 3-repeat isoform, here's the 4-repeat isoform, and you can have a fragment that goes from there to there, or here in the 4-repeat, or here in the 4-repeat. So there are 3 classes of proteins, all bound together within this polymer, the paired helical filament. What these fragments all have in common is that they are out of sync with the normal structure of the repeat domain. So the normal repeat domain has a tubulin binding segment, a linker, another tubulin binding segment. The tau that's in the paired helical filament has a quite different conformation, and we were able to map the N-terminus and the C-terminus of this, and then later went on to show that characteristic N and C-terminal truncation is the hallmark of a pathological interaction between 2 tau molecules. That interaction is self-propagating once it's started. So if we start with the fragment, which is the core paired helical filament building block, it binds with very high affinity to a normal tau molecule, and if you digest that with proteases, what you're left with is simply a photocopy of the fragment you started with, and that retains the ability to bind yet more tau protein. So this is the fundamental basis of the prion-like machine that is driving the progressive aggregation of tau protein. So it forms oligomers, filaments, tangles, and these tangles just go on forming until the neuron just dies. We've recently published modeling this in cell models. So, for instance, if a cell is expressing very low levels of the truncated fragment and you turn on selectively full-length tau, it gets converted into the truncated form as it aggregates, and likewise other cell models. So these are in cells. They're typically 100 units long, so it all happens in quite physiological environment within the cell. What's been alarming in recent years is the discovery that these oligomers, once they form, have the ability to jump into neighboring neurons and initiate the same self-propagating cascade in what were previously healthy neurons. So that's the basis of what's called Braak staging, the neuropathological staging of Alzheimer's disease. So it begins in the medial temporal lobe structures, then spreads into limbic structures and ultimately into isocortex until you get the characteristic distribution of the pathology in the brain, with dense pathology in the medial temporal lobe and then progressive accumulation of tangles. Now, this picture actually came from a 1950s textbook that I inherited when I took over the brain bank in Cambridge. And when we did biochemical studies, we found exactly the same thing. So here is Braak staging shown just as it was published by Braak. And you can see that there is progressive accumulation of tau in the neocortex as patients progress along the Braak stage cascade. Braak staging is highly correlated with cognitive decline. So again, this comes from a prospective study of patients in primary care who were followed until death and taken really at any point in their illness. Whenever they happened to die, we collected the brain tissues. So it was possible to establish a relationship between the mini-mental score in the last 12 to 24 months prior to death and their Braak stage. you can see that what we call MCI, mild cognitive impairment, or prodromal AD is the new term for this, people are typically at BRAC 2 or 3. Mild AD is somewhere between BRAC 3 and 4, and 4 onwards is moderate to severe. BRAC stage is also highly correlated with the functional brain imaging deficits that you can see either by SPECT or by PET scan. So it is really driving a pathology that is impairing neuronal function. So I've explained this part of the process, that the process is self-propagating once it begins. People often ask, well, how does it begin? Probably the most likely factor that's driving the initiation is undigested mitochondrial proteins that overload the endosomal lysosomal processing pathway. And then the APP mutations simply add to that, but are not on the direct causal pathway. Okay, so that's a quick review of the tau pathology of AD. Now I want to talk about methylthioninium, the molecule that we're particularly interested in, and there was the first tau aggregation inhibitor. So we initially started with methylthioninium chloride, so it's the chloride salt of the oxidized form, but we've moved to a stabilized reduced form. So here are the structures, and here the X-ray crystallography. So it's a distinct chemical entity, this reduced form, but that delivers the same active moiety into the blood. We're able to show that these things are active in vitro models. So here is a schematic of an in vitro assay where we have the tau-tau binding interaction, and here we show that it's blocked by an inhibitor. I showed you this model before whereby if you turn on full-length tau, it gets processed into the truncated form. So with increasing quantities of these molecules, of these compounds, you can block that conversion. So it works in cells. And most importantly, if you isolate paired helical filaments from the brain and you add MT, methylthioninium, in various forms, it dissolves the proteolytically stable aggregate and converts it into protease-sensitive monomers. So here are the key results. So it works at a concentration of 0.15 micromolar. The Ki in the cell assay is 0.12 micromolar. So that's about the concentration you need for activity. How does it work? It's not clear at the moment. We're trying to resolve that at the moment. But the kind of candidate mechanism is shown here. The aggregate is a beta-pleated structure that's stabilized by ionic bonds. MT has the ability to disrupt those ionic bonds and as it said, unzip the beta sheet, and then it's released. So it converts from this aggregated form into monomers that are then readily cleared by the cell. So that's how it works with tau. What about synuclein in Parkinson's disease? Well, Parkinson's disease is characterized famously by the presence of Lewy bodies in the substantia nigra and the neocortex, and they're these blob-like aggregates, which are also fibrillar forms of not tau, but synuclein protein. Here is the synuclein, full-length synuclein protein, and here are other proteins that synuclein interacts with. But what's inside the Lewy body is actually a truncated form of the synuclein molecule, and it's that which is the aggregating species. So the model is the same, that alpha-synuclein is produced, but instead of its normal folding structure, the pre-existing aggregates act as a template, and it goes on to form these fibrils, which form the Lewy body. And again, these aggregates, once they form, are infectious via various mechanisms, possibly transsynaptic spread, possibly exocytosis, possibly via exosomes. Nobody is really quite sure. But whatever happens, the process spreads from one neuron to the next and initiates the same prion-like processing in the next neuron. So again, it's interesting that MT, LMTX, or another molecule that we were interested in, you can tune different molecules to have different potencies, that you can demonstrate in vitro assay the disruption of these aggregates with compounds like MT. Here is a cell equivalent assay of the one I showed you before, and you can show the blocking of the aggregates in cells. LMTX is particularly, here is the mechanism, and again it's similar. You've got anti-parallel chains that are stabilized with charge-charge interaction, and these are the beta sheet binding linkages that are hydrogen bonds, and when MT comes into association with this, it disrupts the structure and unravels the beta sheet. LMTX, the molecule that we're interested in, we've shown is active in a synuclein mouse model of pathology, and you can reduce the pathology at 3 months and strikingly at 6 months. So this molecule is perfectly viable to now be tested in humans in Parkinson's disease. And in my second talk that I'll be giving at 11, I'll explain the clinical development of LMTX as it currently is. Similar mechanism with the protein TDP43 in frontotemporal dementia. So here is a case, a SPECT scan, HMPAO SPECT scan of a case with frontotemporal dementia, very characteristic picture. The frontotemporal lobar degeneration syndromes clinically present as a range of syndromes: behavioral variant frontotemporal, semantic dementia, progressive non-fluent aphasia, frontotemporal dementia with motor neuron disease, and of course the FTDP17 cases. All of these, regardless of their clinical presentation, essentially represent mixtures of either tau protein aggregates. I shouldn't say mixtures. They either comprise tau protein aggregates or TDP43 aggregates, typically not both in the same patient. What's interesting with the TDP43 proteinopathies is that the mutations that predispose to TDP43 aggregation are not mutations of the TDP43 protein itself, but in the progranulin pathway, which then acts as a trigger for aggregation of TDP43, which again truncates it, forms aggregates which are toxic and spread from one nerve cell to the next. MT inhibits aggregation in these cells. This is not our work, it's from Yamashita et al., where they made 2 different models of TDP43 aggregating in SY5Y cells, and MT is quite potent at blocking the aggregation, in other words, reducing the number of cells with aggregates. Finally, Huntington's chorea. In Huntington's disease, it involves a movement disorder, but also a disorder of personality and cognitive function, and it's caused by an expansion of the polyglutamine trinucleotide CAG repeat at the C terminus. And in the mutant form, it begins to aggregate, forming cytoplasmic inclusions or nuclear inclusions, which end up in lysosomes. So it's a protein aggregating disease. Again, not our work, but MT inhibits the formation of the oligomers, so and fibrils. So in vitro, it's the same sort of in vitro assay, and again, it's potent at about 1 micromolar in vitro. In mice, a model of Huntington aggregation, you can see that in the presence of treatment of the mutant, of the mice carrying the mutant protein, you can see that at 7 weeks they have improved motor functioning. And in Drosophila fly, in the presence of the mutated protein, there is impairment in the compound eye structure of the fly. And in the present, when you give these flies MT, you produce more of the compound I visual elements, the rhabdomeres, and they are more normal looking. So the toxicity of the aggregate is reduced. So there you are. That's the sort of where the world is at the moment. There was an interesting review by a former colleague of mine in Science last week on exactly what I've been talking about, although what I've been saying is that interestingly, there's a common pathological, molecular pathophysiology going on, but what's even more exciting is that there's potential for a common molecule that treats a whole range of these diseases. So in summary, prion-like processing of aggregating is recognized as a common underlying mechanism of molecular pathogenesis in late life neurodegenerative disorders. It involves a primary conformational change in a critical aggregating domain that becomes self-propagating, forming proteolytically stable oligomers. The oligomers produced in this way are able to infect neighboring neurons, invading previously unaffected brain regions and propagating the pathology. The aggregating domain has a distinct conformation. We're able to show that in tau. We haven't done the work on the other proteins, but presumably it'll be similar. And the methylthioninium moiety has promise for treatment of all these conditions. So as I'll show in my later talk, we are currently doing phase 3 trials in AD and frontotemporal dementia, and in green are the areas that we would wish to explore next. You can see that there's chronic traumatic encephalopathy, which is essentially an aggregating disease of tau protein, and there are all of these movement disorders, progressive supranuclear palsy or corticobasal degeneration, which are tau aggregation diseases, Parkinson's disease, of course, and Huntington's disease. So we are basically seeing a moment of transformation. If our phase 3 results confirm the phase 2 findings in AD, which I'll be talking about later, that will open the doorway to having a molecule that is able to treat all of these diseases. Thank you.